Hello everyone, I am Sutapa MSc in Biology. Welcome to my channel Sutapa's Biology. My today's subtopic under the topic cell is inclusions bodies and cell envelope and its modifications. Cell envelope and its modifications, the subtopic's name. And its modification. Under this subtopic, we shall study about the cell envelope of mainly prokaryote, because eukaryotic organelles and cell envelope etc. we shall study in the eukaryotic cell. But here mainly we shall study about the prokaryotic cell, as the cell wall of the prokaryotes is very complicated thing. They are the very simple organism, but they have the very complicated cell envelope. Now cell envelope, they have three parts. Cell envelope has three parts. Number one, slime layer. Slime layer. It's followed by the cell wall and then cell membrane. So it's a trilaminar structure. It's a trilaminar structure where three layers they have different functions, but their integrated work is to protect the cell. So they have different functions, diverse functions, but togetherly they perform the protection of the cell. That's the main function. Okay, so we are coming to what is slime layer. Now slime layer is a layer which has two types. It has also two types, that is slime layer. When the slime layer is a loose sheath, then it's known as the slime. Known as slime. But if the slime layer is tough and thick, then it's known as capsule. Known as capsule. Now the bacteria which are with capsule are known as encapsulated. So the bacteria, they have two types, either encapsulated, that is they have the capsule, and other is without capsule, non-capsulated. Okay, so either encapsulated or non-capsulated. Now capsule has a important role in this, that with the capsule the cells are very protective in nature. They will not be susceptible to antibiotic attack. So the capsule is good for them and bad for us. Why bad for us? Because antibiotic will not function or will not check their multiplication very easily. So this type of bacteria is known as gram-negative bacteria. Mostly the gram-negative bacteria, they possess the capsule, or they are mostly encapsulated. That's why they are known as smooth. And without capsule, that is non-capsulated, they are known as rough. Okay, so later in Griffith's experiment and etc. we shall study about this. In the Monera also we shall study about this, that what's the role of the capsule in disease producing and non-disease producing. Now next is that, now what is gram positive and gram negative? On the basis of the capsule, they are also divided into gram positive and gram negative. Capsule has the role in this, but main role about this gram positive and gram negative is that the staining property. The gram positive will retain the gram stain. Christian Gram, the microbiologist who made a stain or staining pigment which is known as gram stain. Now gram stain, if applied on the cluster of the bacterial cell, some will retain the gram stain, they are known as gram positive, and some will not retain the gram stain, they are known as gram negative. Now what's the chemical nature of the gram positive and gram negative? Why they can't retain the stain or can retain the stain? Why they can't or can? Now this is the reason that polyriboteichoic acid is a chemical which is present in the gram positive. Polyriboteichoic acid. Mind it, it's complicated name, polyriboteichoic acid is present in the gram positive one, and gram negative they lack this. Polyriboteichoic acid is absent here. Is absent, and that's why they are known as gram negative. Now on the basis of the gram stain, they are divided into two groups. They are divided into two groups on the basis of cell wall and capsule, etc. But they have many different properties. Like that, some group will possess gram positive, possess the mesosome. Gram negative have no mesosome. Gram positive will form endospore. Gram negative have no endospore. So these in details in study in the chapter Monera. Now I am just giving you some examples like this. Okay, next thing is that cell envelope is modified to form some structure. What are the modifications of the cell membrane? Cell wall is the part of the cell membrane which gives the shape of the cell also. Bacterial cell, they have 4 categories mostly. That is, number 1 is coccus. Coccus is a round bacteria. Number 2 is bacillus. Number 3 is vibrio or comma shaped. And number 4 is spirillum. I am giving some rough picture or rough diagram with this, that coccus is like this, round. They might be single, they might be diplococcus, they might be in chain known as streptococcus, they might be in the shape of a bunch of grapes known as staphylococcus. Okay, so these are the types of coccus. The main thing here, the coccus is the round bacteria. And what's about the bacillus? Bacillus is the bacteria which has rod-shaped structure. Either they possess the flagella or not, but they are rod-shaped. But the coccus, they never possess the flagella. Now, what is flagella? I am coming to the next topic, discussion, that what is flagella. Vibrio or comma, that is like this shape, and spirillum this type of structure. So these are the different types of shapes on the basis of the cell wall. Here our topic is the cell envelope, and cell wall is the part of that, and that's the middle part. That's first is capsule, then cell wall, then cell envelope. So cell wall is like this to give the different kinds of shapes to the bacterial cell. Next, what is flagella? In case of the bacterial cell, they are either motile or non-motile. One can move, other can't move. Who can move? They have the flagella. So, motile and non-motile. One can't move, can move, and another can't move. So this categorization or classification is on the basis of their movement, locomotor organs. Now, what are the locomotory organs? In bacterial cells, they have mostly the locomotory organ is flagella. These are the, suppose this is a bacterial cell. This is a bacterial cell. This is the cell membrane. Above this, they have the another layer, that is cell wall. Now, flagella are the extensions of the cell wall. These are the long extensions and they are the extensions of cell wall. So, this is the flagella. And above that, there might be a slime layer or capsule or not. So, this is flagella. In prokaryotic flagella we shall study in eukaryotic cells also. In lower classes you have studied this, that eukaryotic cells they have the flagella also. But their flagella and this flagella is different. Their flagella are made up of microtubule, but here no microtubule is present. The flagella is made up of the protein flagellin. The protein is flagellin. So flagellin protein is flagellin. Easy to recall it, as the name is similar with the flagella. Now, flagella, prokaryotic flagella, they have three parts: basal body, hook and filament. Three parts. So, flagella can be divided into three parts. You will get in NCERT book also that flagella of prokaryotes. They have three parts, that is basal body, hook and filament. So by the movement of the flagella, the cell can move. Now the another extension, these are the extension of the cell wall, and another extension is fimbriae or pili. Fimbriae or pili is just like this. They are more in number in a cell than the flagella, and they look like this. They are the smaller appendage and hollow. They are smaller and hollow appendage. They are known as pili. They are also made up of pilin protein. Pilin protein, like the flagellin. So these all are made up of protein, different types of protein. So this is pilin protein, and their main function is to in case of the parasite, they just clean the host body with the help of the pili or fimbriae. And in case of the bacterial reproduction, sexual reproduction, they take part in the conjugation. And the cell which possess the plasmid of F factor, they can produce the pili for the sexual reproduction conjugation. So this is about the pili and fimbriae and flagellin. Now in the next part, we shall study about that the another inclusions, these are external, but the another one, that is the part which form internally, that is the plasma membrane is here. It's very easy to think that inclusions in infoldings will be from plasma membrane. As cell wall, if from the infolding, then it has to pass through the cell membrane. It's not possible. Practically, it's not possible. So that's why in case of the infoldings, they are arising from the cell membrane. Okay. Now in cell membrane, you see that suppose this is the cell membrane. Now it has cell wall above it. And another is slime layer. It might be present in a slime or capsule. So this is the thing. Now infolding of this cell membrane, it's forming the mesosome. Now what is the function of the mesosome? Mesosome is a coiled structure and it's remain very near to the genophore. I am giving the rough diagram of the genophore. Suppose this is the genophore, then mesosome is here. This is mesosome. Now what is the function of the mesosome? Mesosome has three parts: vesicles, tubules, and lamellae. These three parts, vesicles, tubules, and lamellae, they form the cell wall. They help to form the cell wall. And another, they are very important for the performance of the cell respiration. In mesosome is you will study about that. The mesosome is the analogous structure of the mitochondria. Like their origin is different, but they are analogous with the mitochondria. Their function is same. Mesosome perform the cell respiration. Mitochondria also perform the cell respiration in higher organisms. Okay, so mesosome is just like the mitochondria of the eukaryotic cell. Now, mesosome has three parts. They form the cell wall. They take part in the cell wall formation. Another, they help in the replication of the DNA, as they are very close to the DNA. Genophore is the DNA of the bacteria. So they are residing very close to the genophore and help them to replicate. And next thing is that they help in respiration also. Now we have another thing to discuss, that is what's about the cell membrane. Cell membrane of the prokaryote. It's very important to know that cell membrane, other things are different than the prokaryotes. That's why they are placed in the eukaryote due to difference of them with the eukaryotes. But in case of the cell envelope, they are just like the other cells. That is the PLP structure. What is PLP? PLP is the protein, lipid and protein structure. Protein, lipid, protein, and we shall discuss this about this structure in details in the cell membrane part. In the next part, there is the cell membrane, where we shall discuss about the PLP. So in case of the eukaryotic cell, they have the PLP and protein, lipid, protein. That means two layers of protein. In between that, the lipid is sandwiched. Now you just know this. Later we shall study about it, about the locations or distributions of the protein over the lipid, how it's possible, or what is the justified one. Now here PLP structure is also present in bacteria. So bacteria has the same cell membrane like the eukaryotic cells, and cell membrane is semi-permeable. Like the others, their cell membrane is also semi-permeable. What is semi-permeable? That means they will not allow all the things to go inside or come out. Whereas cell wall is the permeable one, through which there is free movement of the materials, molecules, ions, etc. So that's all in this topic today.